So it's my pleasure to introduce today's Grand Rounds speaker, Dr. Luis Diaz. Dr. Diaz is an associate professor of oncology at Johns Hopkins Cancer Center, and he specializes as an oncologist in the treatment of pancreatic and colorectal cancers. He received his undergraduate and medical degrees at the University of Michigan in Ann Arbor, and then he completed his residency training at the Osler Medical Service at Johns Hopkins. His medical oncology training was also done at Johns Hopkins in the Sidney Kimmel Cancer Center, and then he did his fellowship research in Bert Vogelstein's laboratory at Hopkins. In under a decade, Dr. Diaz has pioneered several genomic diagnostic and therapeutic approaches for cancer. He's a member of the Ludwig Center for Cancer Genetics and Therapeutics, where he directs the translational medicine research. In particular, his work focuses on clinical development of tumor-derived DNA as a biomarker for cancer screening, early detection, monitoring, and measurement of residual disease. So today's lecture will be on the clinical application of cancer genetics, a molecular pap smear. This technique is really a promising approach for the early detection of ovarian cancer and endometrial cancer, and he's going to go and also tell us about his other work in the Circulating Tumor DNA, which just appeared last week in Science Medicine?
Science
Translational Medicine. A year ago, when all his work on this ovarian cancer and endometrial cancer early detection came out in the PAP test, this was really highlighted in the New York Times and NPR, CNN, and NBC Nightly News, so you may have heard about this. So it's a great pleasure today to have Dr. Diaz here to tell us about his research and also to experience the polar vortex of 2015. So there you go.
Thanks, Amy, and thanks for the invitation to come to speak to you. Originally, I was going to speak about the molecular pap test, but I thought that more useful would be the journey that we took to get there, because there is a lot of molecular biology and cancer genetics behind that hypothesis and eventual implementation or pilot study that took place in order to better understand how to do that. So I do have some disclosures. Founder of a company, PagerBox, in '99. This is pre-Facebook and pre-WhatsApp. It was a text messaging service, so doctors and nurses could text message one another. I started at Hopkins, and the only other place that really took off was University of California Healthcare Systems. Hopkins abandoned it about 2 years ago. I haven't changed the code in a decade, and they still use it at University of California Healthcare Systems. You'd think they'd be more modern or advanced, but that's still ongoing. Founder of a company named Personal Genome Diagnostics and consultant for NRO Pharma and Amgen. So I'm going to talk initially about cancer genetics. And really over the past decade, we've seen an explosion in the knowledge about cancer genomics. And really that's been fueled by sequencing technology. Sequencing technology moved from rudimentary Sanger or PCR pyro-based sequencing to next generation sequencing. And what that's allowed is whole exome or whole genome or gene family analysis across many tumor types. And this is just a picture that depicts all the different tumor types that have been looked at. And these are exome studies, so all the coding regions in the genome, about 20,000 genes, where they've looked at the tumor DNA and compared that to the matched normal. And that's very important because that's how we define mutations. Somatic mutations in the tumor are present only in the tumor and not in normal. And that's important as we move on to the diagnostic. And what you see when you look at all these different genome studies is that cancers are all not the same. Pediatric tumors have very few what we call point mutations. Liquid tumors, hematologic tumors have very few point mutations. Adult solid tumors or sporadic tumors have a smattering of usually less than 100 mutations, but more than 20 per tumor. And then you get the mutagen-associated cancers, lung cancer, melanoma, lung cancer from smoking, melanoma from UV light, and you start getting into the hundreds. And then you have things that are diseases associated with lack of DNA repair, hereditary syndromes. In this case, MSI or Lynch syndrome, and you get into the thousands of mutations per tumor. So there's a wide spectrum of mutations. And one of my goals when I joined the Vogelstein lab as a postdoc was, how do we take this genomic information and use it clinically? And I'll walk through how we got to using these as diagnostic markers. Point mutations, which were on the previous slide here, are only one type of genomic alteration that are somatic. You can also get translocations, the most famous one, BCR-ABL, deletions, amplifications, again another famous one is MYC or HER2-neu, small insertion or deletions, and then single base pairs. But you can see that the majority of alterations in these tumor types, from breast, colon, pancreatic, glioblastoma, are point mutations, and the minority or the rare variants are these other types of genomic alterations. So how do we take this and apply it as a diagnostic test? And the model system that I began working on was on circulating tumor DNA. And using these mutations as a way to diagnose cancers and to apply this to difficult clinical questions. But I'm going to begin with the biology of circulating tumor DNA because it's not an area that a lot of people know about, and it really began primarily in basic science work that was clinically applied to fetal medicine, looking at circulating fragments of the fetus in the mother's blood circulation, where you can look at genetic traits of that fetus just simply by a blood draw. And the circulating fetal DNA is very abundant. It's about 10% of the total DNA fraction in the mother's blood as early as 10 weeks after conception. The question is, could we do the same thing with tumor DNA emitted and released from tumors into the circulation? And this is the basic principle that really allowed this to go forward. These mutations, how they're defined in the tissue, make them very specific. I like to say perfectly specific. They're only found in the cancer or precancerous tissue, but not found in matched normal tissue. So from the get-go, it's the type of system that would be minimal false positives, because these were defined as being only in the tumor and not in the normal. And this was one of the initial principles that really led this project forward. The second is that when you draw blood from the patient, and whether it's serum or plasma, and you isolate DNA, that DNA is, in a cancer patient, mixed. The DNA is normal versus tumor. And mutations or cancer genomic alterations lend themselves nicely into differentiating these two fractions. Here, the normal is wild-type DNA or unmutated DNA, and the tumor fraction is going to be mutated. So two concepts, highly specific, and you can differentiate the normal DNA from the tumor DNA in the circulation by mutations. However, there was a problem. Here I represent this fraction as a pretty chunky fraction. But circulating tumor DNA is actually pretty rare. It exists at a level of typically less than 1%. And that's not detectable by standard Sanger sequencing, pyrosequencing, or even real-time PCR, primarily because of the background that's generated is above 1%. Not because these aren't sensitive enough to detect them, but because you introduce errors, the polymerase introduces errors to the point where you can't see below 1%. So there were digital-based technologies that could actually look at individual molecules and determine whether or not mutations were present at a sensitivity of less than 1.1% or even 0.01%. And we set the floor at 0.01% because that is the error rate of the polymerase. So even if you're looking at an individual molecule, in general, you're going to introduce errors at that rate. And Devin Dressman in our lab, who's actually now at Life Technologies, developed a digital approach in our laboratory to look at this exact issue, and it was called beaming. And the technology was so forth. So in a complex mixture of DNA, tumor and wild-type DNA, let's say we're looking for KRAS in this situation, you amplify the region of interest. And let's say codon 1 of KRAS if you're looking for changes in codons 12 and 13. And you preamplify this region of interest, and then you mix these amplified DNAs in a mixture of oil, water, and magnetic beads. Now when you amplified these, you also added a linker on both ends, and that linker was complementary to sequence here coding the bead. and you created a slurry, and that slurry would naturally become bubbles at a ratio where one bubble would contain, on average, one bead and one template DNA molecule that would bind to the surface of that bubble. Now, the aqueous solution in this water-oil mixture is PCR buffer. So you create billions of bubbles, each compartmentalized, at a ratio where you're going to have one fragment of DNA or template DNA, one magnetic bead, and you apply these PCR to this. And you coat the entire bead with the same strand that was in that bubble. And with allele-specific hybridization, or single base pair extension here, you can differentiate mutation from wild type. And when you pop the bubble, and this allele-specific hybridization is done using fluorescence, you can differentiate these using flow cytometry. So here you have the wild type population, and here's the mutant population. Now, what I described is the perfect scenario, where you have one template molecule and one bead per bubble. But you do get situations where you have two template molecules per bubble, two beads per bubble, no template molecule and two beads or one bead, and that's what we see here and here. So it's not the majority, but that's probably because of the efficiency of PCR. It's more efficient when you have one template molecule per bubble, and you can differentiate the wild type from mutant using allele-specific hybridization in this case. And this is an example where we looked at a specific KRAS mutation that was 100% abundant, and then diluted it down to 0.05%. Now you may ask, if it's 100%, why do you still see wild type? Well, there's two things. One is KRAS mutations is in one of two alleles. And number two, the wild type efficiency of PCR in this case is a little bit better. And the sensitivity or detection rate in this case, and in many cases, was at least 1 to 10,000 to 1 to 500,000. And what this allowed us to do was look at some clinical scenarios in a way that we hadn't been able to before. And this is an example of monitoring disease by looking at circulating tumor DNA. And this is a patient who we knew had a mutation in a gene called APC, commonly mutated in colon cancer. So we knew the mutation down to the base pair, and we created a beaming assay, one of these assays, specific for this mutant APC mutation. And this is that flow cytometric histogram that I showed initially. And this is the APC mutation in this patient before he underwent a surgery. This is the wild type APC. This is the fraction that I talked about, having two template molecules or two bubbles and or two beads and one template molecule. And this is the mutant population. And this patient was undergoing potentially curative surgery. We drew their blood one day after surgery, and you can see that the majority of circulating tumor DNA is gone, but it's not completely gone. At day 42, they got a CT scan. No tumor was visible, but you start seeing a slight increase in the amount of circulating tumor DNA. And then at day 244, the CT scan is finally positive, and you see a more robust signal from the circulating tumor DNA. So kind of a way to non-invasively track the viral or the tumor load, similar like a viral load in HIV. So could you see the fluctuations and the dynamics in this patient's clinical history, and would those match what we see in the circulating tumor DNA fraction? And that, I'll pause there and actually step back. So what are other things that we know about these circulating tumor DNAs? Well, we know they're very small. This is a study from Stephen Quake, not in cancer patients, but in normal patients, and this is actually a specific patient with who's pregnant. And they did next generation sequencing across all the DNA in the blood, and they found that most fragments were about 180 base pairs long, which is the exact length that wraps around the nucleosomes. And these are the fragments that actually end up entering the circulation after apoptosis. And if you do this on a gel, you actually see ladder at 180, 360, and so forth, all that's consistent with the apoptotic ladder. And the other thing we know about these fragments is that they're dynamic. They change with surgery. This is not tumor DNA, but total circulating DNA. And you can see the fluctuations with surgery, with chemotherapy, with another surgery, with chemotherapy. This is actually a real patient. And then when you look at tumor DNA, when you're looking at specific mutation here in PI3 kinase, you see these fluctuations as well. And then finally, these fragments have a very short half-life. They have a half-life of about 2 hours. And this is true for fetal DNA. This is true for circulating tumor DNA. They don't last a long time. And this is an example of one of many patients where we saw the circulating tumor DNA disappear over 2 hours. And the half-life was about 2 hours. We saw it disappear over a time, quite acutely initially, and then more drawn out over time. So we were quite excited after these pilot studies. So this circulating tumor DNA maybe could be used like a viral load. And could we use this to answer some clinical questions that we couldn't answer in any other way? And the basic model was, when we did this, patient has a resection, we sequence the tumor, and then we create a specific beaming assay. And most recently, and this is the paper that was just published, it's no longer in press, we did this on 14 different tumor types, from bladder cancer, colorectal cancer, all the way to brain cancers. And not surprisingly, not all tumor types release the same type of circulating tumor DNA into circulation. Tumors like bladder, colon, esophageal, ovarian cancers have high levels in the circulation, whereas tumors like gliomas or brain tumors have very low levels. And for some reason, tumors like renal cell carcinoma and thyroid cancers, these are all metastatic, have very low levels as well. And we don't fully understand why. Some of it might be locational. Some of it may be the rate of division of these tumors. But some of these tumors down here were fairly aggressive tumors, so we're not entirely sure. The one thing that is clear is barriers, like the blood-brain barrier, doesn't let these fragments leave the blood circulation. And I suspect that means that these fragments can't get into the brain circulation either. And in some cases where we couldn't detect these tumors, they were tumors with histopathologic features like mucinous features. So mucin coating the tumor, that may limit the amount of circulating tumor DNA that can enter the circulation.
Quick question on that.
Yeah.
Are you able to correlate that with either lymphatic or vascular cancer?
So whenever we've looked at vascular invasion or lymphatic invasion, differentiation status.
Just density in the tumor as far as, tumors that tend to be more vascular.
Yeah, we actually specifically looked at,
in the absence of true invasion.
we looked at that in pancreatic cancer, and we found no correlation with that, because we thought that would be the obvious one.
Yeah.
Then I thought maybe necrosis would be another obvious one, and it wasn't.
Yeah. Fairly vascular too, which is why.
Yeah, renal cell carcinoma is incredibly vascular.
Yeah, so it doesn't completely explain it. And a lot of our work was in pancreatic cancer, and we thought pancreatic cancer, these metastatic patients had disease everywhere. It was very low. So there's some other biological feature going on. One question I always had is there an increased amount of circulating DNases that degrade these faster? Because the half-life studies that we did were in colorectal cancer patients. We haven't done them in these other patients, and they're a little more challenging. And this is just the distribution of the circulating tumor DNA in these different tumor types. So wide distribution, really patient-specific. And this is just a simple graph that shows that survival is worse as the number of fragments increase in the circulation. And this is across 120 different metastatic colorectal cancers. So in terms of biology and just what I've introduced you to at this point, these fragments, these circulating tumor DNA fragments are highly specific. Again, based on how we define mutations in the tumor, but not in normal. They're very small. They're 180 base pairs. So oftentimes I'll review papers where they've amplified amplicons that are 300 base pairs or too big to capture these small fragments. And that's why those papers oftentimes don't have the sensitivity that we like to see. They're dynamic. You see changes with the clinical course of the patient, with surgery, with chemotherapy, with healing. Very short half-life, at least in the colon cancer patients, 2 hours. Remains to be seen in other tumor types. Heterogeneous across tumor types, and levels correlate with survival. What about beyond point mutations? Over the past 10 years now, we've looked at other things. Mung Lee, who's actually a pathology resident now, performed methyl beaming and simply doing a bisulfite conversion of the DNA prior to applying the beaming technology, and then we were able to differentiate the unmethylated versus the methylated regions to look at methylation status. Less specific than mutations, because you can get methylation in normal tissue. You can get age-associated methylation that looks like cancer. You can get environmental exposure methylation that's not cancer that looks like cancer. But nonetheless, a technique that's important because methylation is important in many disease types. This is work by Yiping He and Nick Papadopoulos, where they looked at mitochondrial mutations. And this was of high interest to me because in most genomes, there's one mutation per genome. So that would give you one fragment from each cell to track in the blood. If you had a mitochondrial mutation, you have 10,000 or more mitochondrial genomes per cell, so it would be incredibly sensitive. And this is an example. This is a patient with an APC mutation in their genomes, 8 and 9, and patients with mitochondrial mutations. You can see here the APC mutation, in terms of mutant fragments per mil, is quite low, whereas the mitochondrial mutation is quite high. Two issues with this approach. Mutations in the mitochondrial genome are rare, and when they occur, they're typically not what we consider homoplasmic. The mitochondria are all not homogeneous throughout the same cell. They're heterogeneous, they're heteroplasmic. So that's the one barrier to really going forward with that approach. More recently, Rebecca Leary and Victor Vaculescu looked at rearrangements in the blood. And this was actually a funny story. Initially, Rebecca was doing her postdoc on looking for new rearrangements and translocations in different tumor types. So it was a very long graduate student PhD. She was at year 5, and she had done whole genome sequencing on probably 40 breast cancers and 40 colon cancers, and she had found no specific mutations or genomic alterations. In fact, every tumor had a rearrangement, but it wasn't common to all the other tumors, so there was no repetitive rearrangement to find, and there was really nothing to report. So by chance, we had plasma on these same patients, because I'd been collecting plasma. And so the question was, could you look at the rearrangement you found in the tumor in the blood? And this paper showed that we actually could, and you could actually monitor those rearrangements in the blood. They really took this to the next level, and they started looking not at just rearrangements, but aneuploidy, chromosomal gains, chromosomal losses, deletions, in addition to amplifications, translocations, and normal changes. And what they were able to see were chromosomal gains and losses in the plasma that corresponded to those in the tumor. And let me get to there. I'll come back to the other figure in a second. But this is normal plasma. This is a tumor from a patient, and this is plasma from a patient. And you can see that the chromosomal gains and the losses match from the primary tumor to the plasma. Now, this patient underwent surgery, and the level of circulating tumor DNA went down quite low, and that's why you lose the resolution that you had here when the percentage was 37%. But when the patient recurred again, you see the same sort of molecular changes or genetic changes that you saw in the primary tumor. The other thing that was possible from these whole genome approaches was we're looking for translocations, and even specific translocations that were actually amplifications. And this is something that we began to learn as we dug deeper into this, were that amplifications by nature are novel translocations. So when we were looking for translocations, an amplification in ERB2 or HER2/neu actually genetically was defined by a new translocation. So you could find that by looking for these nouveau genes. So I talked a little bit about the biology of these beyond point mutations, and then I'm going to move on to some clinical applications. So what I've described are these breast cancers or pancreatic cancers or colon cancers that as they turn over, as they undergo normal apoptosis or even necrosis, they will release fragments of DNA into the circulation. What can we do with these fragments that will have a clinical impact? Not just something where we can monitor tumor change over time, but something that will change the outcome of the patient. And typically, someone who focuses on diagnostics, it's a challenge to make a diagnostic as potent as a therapeutic intervention. But that's really the goal that we're trying to do here. And the first example I'm going to give is tracking resistance. Currently in cancer, when we apply a targeted therapy, in a fraction of patients we'll see a response. But invariably, the patient becomes resistant to therapy over time while they're receiving that targeted therapy. Could we use this technology to look for resistance mutations over time in the circulating tumor DNA fraction that corresponded to that resistant clone? And our colleagues in infectious diseases have been doing this all the time, either with HIV viral load, looking for resistance mutations over time. Even from blood cultures, you can follow resistance from bacteria over time. And our hope was, could we do this non-invasively? And we picked a model system where we didn't fully understand the mechanism of acquired or secondary resistance. So we looked at EGFR blockade in colon cancer. And what we knew was that if you had these mutations in KRAS, you don't respond to this therapy in colon cancer. It's well known, and it's a FDA-approved test now, and on the package insert for this drug, telling you that you have to check for these mutations before you treat with this. So if you're wild type in these genes, the response rate is about 17%, but that response only lasts about 12 weeks. So we decided to look at a variety of mutations in the KRAS pathway in the plasma to see if we could define the molecular resistance profile of patients treated with EGFR. And what we found was that in patients who were initially wild type, treated with anti-EGFR antibodies, over time, that we could see the emergence of KRAS mutations. And these are patients that initially responded and then became resistant. And here's an example of a patient who was wild type in KRAS, but then you saw the emergence of 4 different KRAS mutations in that patient. And more recently, we looked at a variety of other mutations, and we see these patients who are initially wild type for all these genes become mutant in many of these genes. And all of them had more than one mutation. And I'll move one slide forward. So patients who initially KRAS wild type and actually wild type for all these genes becomes mutant in a variety of different genes in response to the selective pressure. And it sets up a scary picture, because this patient you thought you could treat with one targeted therapy is now heterogeneously mutant in a variety of different genes because of the selective pressure. How do you treat this patient? He's already resistant to EGFR blockade. There aren't enough drugs right now to treat for all those different types of mutations. So it sets up a big challenge. But what we found from that initial study is that about a third of those patients develop resistance to EGFR blockade by the emergence of KRAS mutations. As I showed you, there was metastatic heterogeneity or resistant heterogeneity. And probably one of the most important features of the study was, so if we had biopsied every tumor in every patient in the study, we would have done 1,100 biopsies over time. Not feasible. So by drawing the blood, that was a collection of all the mutations from all the tumors in that patient's body, and it gave us a glimpse at the heterogeneity. I missed the title here, but so that was one, I think, clinical application that is more futuristic. So as we use more targeted therapies, we're going to be able to see the mutational profile of the tumors as they evolve, and maybe, just maybe, we can target drugs a little bit more better, or mix drugs in an appropriate fashion to prevent this from happening. This is another topic that I want to get to, and there was a title slide before this. I'm not sure what happened to it, but one of the areas that had always frustrated me as a clinician was, after a patient went to potential curative surgery, the surgeon would say, "Oh, I got it all." And then I would be stuck telling the patient, you know what, you may not be cured. We have to wait 5 years to really figure this out. Is there a way to determine whether the patient's cured or not immediately after surgery? And we took 18 patients undergoing potentially curative surgery with colon cancer. And after the surgery, we took their tumor, sequenced it for the known mutations. These were all colon cancer patients. And we created a personalized assay. And we also drew blood 6 to 8 weeks after surgery. We drew blood 1 week after surgery, and we drew blood 1 day after surgery. And because colon cancer has a well-known biomarker, CEA, which is a protein, we also checked that. And this is the 6 to 8 week time period after surgery. And what you can see here is CEA, the protein biomarker, like CA125 or PSA, after surgery was undetectable in these patients and detectable in these patients. Now, if you had detectable levels, your prognosis was worse, and I can tell you this is statistically significant. But this isn't clinically satisfying. If you're a patient on that curve, you're not going to feel wholly reassured. But certainly if you're a patient on that curve, you should probably undergo more aggressive therapy, but that's not our practice currently. When we looked at circulating tumor DNA after the surgery, so in patients where we took their tumor, sequenced the tumor, made a beaming assay specifically for the mutation in their tumor, we found all the patients with detectable circulating tumor DNA 6 to 8 weeks after surgery recurred, whereas those patients with undetectable levels, where we couldn't detect circulating tumor DNA, were cured. And we followed these patients out for over a year.
What percentage of patients had undetectable tumor DNA?
So this is 4 patients.
Out of how many?
This was about 18. And the patients we picked here were patients with metastatic disease, so getting metastectomies, typically of their liver. And those patients typically have a cure rate after metastectomy of about 20 to 30%. So this is representative, maybe a little more aggressive because we have some aggressive surgeons. But it reflected the potential of using this as a way to tell patients whether they're cured or not within a time frame where decisions about adjuvant therapy and postoperative therapy could be used. We're currently looking at this in breast and in earlier stage colon cancer, and I'm hoping that the results look somewhat similar to this. The one exciting feature is when we look back at 7 days and even 1 day after surgery, the figure looks pretty much the same. We did miss a couple of these down here, probably because the half-life of circulating tumor DNA hadn't allowed for regrowth of the tumor at that point. So 6 to 8 weeks may be the sweet spot. But it would be neat to be able to tell the patient before they left the hospital, hey, you're in this group, we need to treat you aggressively, or hey, you're in this group, let's just treat you with the standard of care, which is a more attenuated chemotherapy. Currently, everybody gets the same attenuated chemotherapy because you don't want to overtreat these patients and you don't want to undertreat these patients. This would allow us to really focus and treat more aggressively the patients with detectable levels. And I'm going to wrap up with what I think is probably the most important application, which is early detection, and which will finally get us to how the Pap smear studies really emerged. Early detection is an area where I think that as a treating oncologist, where I see a lot of futility in some of the therapies that we use, can have a real impact, especially for the sporadic cancers like colon cancer, breast cancer, pancreatic cancer, ovarian cancer. And in order to see the feasibility of using circulating tumor DNA for early detection, we use the same approach. And we took tumor tissue from patients across many different stages, across 15 different tumor types, and looked for mutations in the primary tumor tissue, and then looked for the mutations in the corresponding plasma from those patients before they underwent surgery. And this is a figure from the recent paper where we looked at a variety of different tumors, but this is a snapshot of colon cancer, gastroesophageal cancer, pancreatic cancer, and breast cancer. As you can see, you can detect all metastatic colon cancer, the majority of gastroesophageal cancers, many of the pancreatic cancers and breast cancers. But in early stage, which is stage 1, 2, and 3, we could detect 75% of the colon cancers, 50% of the gastroesophageal cancers, a little under 50% of the pancreatic cancers, and about 50% of the breast cancers. When we looked across stages, and this is inclusive of the tumor types beyond colon cancer, gastroesophageal, breast, and pancreas, we saw a similar distribution. When you get to the metastatic level, you can detect many of them. And when you get to the stage 1 level, where typically 90% of patients are curable, you could detect a little bit over 40%. And the distributions are shown here. So this set up a very exciting possibility for us. But one issue was that getting from 40% to 100% or even 90% was going to be a challenge. Were these the biological limits of circulating tumor DNA? And were there other ways that we could potentially use this same technology to detect cancer earlier? And the way that we thought that these fragments entered the circulation was that as the cell was undergoing necrosis, or as the cell was entering the circulation, or as the cell was undergoing apoptosis, these small fragments would enter the peritumoral fluid and then be captured by the lymphatic flow or into the circulation. But what about other peritumoral fluids that collect these fragments as well as circulating cells? And one hypothesis we had was, could we apply this to ovarian and endometrial cancer? And could we apply it in such a way that the ambient fluid around an ovarian cancer or the ambient fluid around the endometrial cancer would collect these fragments, collect cells, and would the cell, rather than entering the peritoneum, also enter the fallopian tube, migrate through the uterus, and lodge in the cervix, and be collected by a pap smear brush? Would it survive that transit? And what were the chances that we could capture a cell? And likewise, could an endometrial cancer cell slough off and migrate through the uterus, survive that transit, and lodge into the cervix? And at the time of a routine pap smear, could we detect those cells or mutations from these tumors? And so what we knew was that this disease is not incredibly common, but the deaths were very high from ovarian cancer and not so high for endometrial cancer, but certainly something where no screening tests existed. None exist whatsoever currently. There are no recommendations from any guiding bodies. So what we did was we harvested DNA from standard pap smears. These were pap smears in women with suspected endometrial and ovarian cancer. And we interrogated 46 different gene regions and genes commonly mutated in endometrial cancers, and we applied a technique called SafeSeq. So moving away from beaming to another technique called SafeSeq that uses next-generation sequencing to interrogate multiple different regions of interest in the genome. And Isaac Kinde really helped pioneer this technique. We were talking before that the length of our graduate student training, he was in the lab for 7 years. And for the first 5, he had no publications. He started to get depressed around 4.5 years, but things started to pop around year 5. And the approach that he used was called SafeSeq, so safe sequencing. And initially we thought, okay, next generation sequencing is here. We can apply the DNA right to the machine, sequence very deeply, and find the mutations. The problem is, similar to Sanger sequencing or real-time PCR, there's an intrinsic error rate to next generation sequencing, and it's not low. It's about 2%. So you're going to miss a large number of rare events. So what Isaac did was look at assigning barcodes to each fragment in the complex mixture. And what that set up was a situation where if there was a mutation that was real, you'd have a jackpot in the barcodes in those mutations. But if you had mutations that were spurious or introduced by the machine, you'd have a low level of mutations, and you wouldn't have jackpots in that mutation because you have many repetitive reads. And because of the genome sequencing project, we were able to pick the regions of interest for the most common subtypes of ovarian and endometrial cancer. And this is endometrial cancer, and we knew the spectrum of endometrioid, serous endometrial, and clear cell endometrial. and likewise in ovarian cancer, we were able to pick the most commonly mutated regions for each of these tumor types. And going back to the principle, so once again, do cells or DNA make it to the cervix, and can they be collected by the pap smear brush? Or endometrial cancers, do they lodge in the cells lodge in the cervix and can likewise be collected in the brush? Could we isolate DNA from this brush and then apply the SafeSeq technology? And what we found, surprisingly or not surprisingly, was that in endometrial cancer, we could detect 100% of the cases, and even very early stage cases. And in ovarian cancer, we could detect about 40% of the cases, most of them advanced, but a handful early. And these tumors that were missed in the ovarian cohort, we knew what mutation to look for. We knew that our panel contained the mutations that they had in the tumor tissue. So these weren't missed because we looked for the wrong mutation. They were missed because biologically, and maybe because of the sensitivity of this technique, the cells didn't make it, or the DNA didn't make it to the cervix for collection by the pap smear brush. So to summarize the results here, we detected 100% of endometrial cancers, 40% of ovarian cancers, and in an age-matched cohort of healthy controls, we did not have any false positives. Again, harking back to the definition of mutations, only in the tumor or precancerous tissue and not in normal tissue. And we're currently conducting future studies to validate this approach, not only in repeating the sporadic endometrial and ovarian cancers in a larger cohort of 500 patients, but also looking at high-risk population with Lynch and BRCA carriers. Subsequent to this, we've done other studies looking at other methods to collect DNA from the cervix or from the GYN tract in a better way. In collaboration with researchers at University of Alabama, we had tampons placed in women with suspected ovarian and endometrial cancer, and we looked to see if the tampon, because it's collecting fluid over a 6 to 8 hour period, could be used to collect cells. Subsequently, we took that DNA and looked at it, and we haven't looked at the endometrial cases yet, but in the ovarian cancer cases, the sensitivity is similar, about 40%. But then there was a very clever pathology fellow who actually went back and read the pathology reports, which we hadn't in the cases, and the women who we missed had tubal ligations. So just maybe we're missing some of these cases as well because of tubal ligation, which makes anatomical sense. And we'll see, but it's the simple things, right? It's the simple things and it's the common sense things that I think are going to play a big role in this. So to summarize, I think that somatic mutations, beyond what we all think their primary goal is, which is to be a target for therapy, can be used as effective diagnostic markers that can have a big impact on clinical care. And that's something that I've really tried to push, is answering questions that were difficult to answer, that couldn't be answered in any other way using this technology. And these mutations can largely be successful because of their specificity, whether it's in the blood, other fluids like the pap smear. Digital genomics and next generation sequencing has improved the sensitivity, not because we can detect the mutation better, but because there's so many events, we can lower the error rate, or we can see the error rate is a better way to say that. And the throughput is sufficient now for real clinical application. I think this will be coming, and I mean, there's a lot of players out there trying to get this in the clinical setting with a turnkey solution for looking at these alterations. And I really think, and this goes beyond just this talk and this topic, is that what we've been trying to focus on unmet clinical needs. And we focused a lot on the technology early on, and we thought the technology was really cool, but really to transform and translate this from the research into the clinic, we've got to focus on questions that will really impact clinical care. And that's what we've tried to focus on, and that's what I've tried to show you examples of today. And like I said, I think early detection, even though oftentimes it's not the sexiest topic, I think it can be a very high impact topic in the future, especially if it's done correctly. And then finally, what I wanted to talk about was the evolution in the technology and how I think things will change in the future. Initially, the beaming approach that I described was looking at single point mutations. Next, the safe sequencing approach looked at regions of interest. The approach that Victor and Rebecca looked at structural changes and even exome changes in the genome. And more recently, we've tried to do whole genome changes. So from a targeted approach, a very targeted approach, to a more unbiased approach. And currently, this costs a few dollars in plasma. This costs $50,000 per patient per plasma. But I can assure you that as the cost of sequencing drops, because these costs are all related to the cost of sequencing right now, we'll be able to do more and more of these studies and see more and more of these changes in rare variants, as in tumors, as in plasma, as in pap smears. And this is where I'm seeing the field technologically moving towards. And more the sequencing companies are moving to decrease the cost of sequencing and allowing higher throughput in terms of sequencing. I'm going to stop there. I'll take any questions, but before I stop, I want to thank my mentors and colleagues, Bert Vogelstein, Ken Kinsler, Nick Papadopoulos, Victor Valkulescu, and Shibin Zhao. I couldn't have a better group of mentors and colleagues to work with on these studies. And as we were talking before, more and more, late at night, I see the faculty left in the lab, and all the trainees are gone. And I'm seeing a shift, and I don't know if that's true here, too, where the faculty are doing more and more work, and the trainees are doing more focused work. The other group I want to thank is Swim Across America. No one would support the PAP gene study, whether it was big funding sources, specific funding sources. But there's a group of swimmers, 500 swimmers, who jump in the Chesapeake Bay each year and swim 1 mile to 3 miles. And they usually pick 1 or 2 groups to help support, and they were the pilot funding for that research. So I have them to thank for, because no one believed in these swimmers in the beginning. So with that, thank you very much. Happy to take any questions.
Questions?
In the cases where you showed metastatic heterogeneity,
Right.
it wasn't quite clear how sensitive the circulating tumor DNA was to pick up the different types of heterogeneity. Can you address that?
Yeah. Let's go back to those figures. So this is a depiction of what I think is happening in the tumor, because we know these mutations emerge. I can't answer your question, because in order to answer that question, I have to know the mutation. This, let's say, mutation is in that tumor, and I don't know if we missed other mutations in other tumors. We'd have to essentially do either a biopsy or do this at warm autopsy to really define the lower limit of detection in the resistant setting. One could do it potentially by continuing to follow the patients and interrogating many mutations and see when the mutations emerge and try to predict which corresponding lesion on the CT scan corresponds to that mutation. Victor Valkulescu is currently doing studies where there's a warm autopsy program at Hopkins where they're actually taking all the tumors in the patient's body, sequencing them, and trying to match it with the plasma levels to try to answer that question. But to absolutely answer that question, it's impossible. So what this gives you here is a sense of what's going on. I can't say with confidence that it's detecting all the mutations with high sensitivity, because there's even a heterogeneity in the amount of tumors and clones in that patient's body. Yep.
Two questions. One, when you sequence the tumor initially, are you doing it off fresh tissue or?
So we've done it on both. We can't decide sometimes because that's what's given to us. I prefer to do fresh tissue because it's better. I like paraffin because we can do it in the lab and I don't have to involve the pathologist all the time. But when you have frozen, you actually have to cut it through, and it's a little bit slower.
And then question two, you're targeting, it sounds like, or it seems like, mostly DNA. Are you targeting RNA in your circulating?
So we've tried both messenger RNA, and that's been very challenging, primarily because of reproducibility. We can take the same sample, crack it in half, isolate the RNA, and either in a targeted approach or in a genome-wide approach, look at RNA expression, or even tags, and the two samples won't match up. And I think that some of it has to do with the half-life of RNA, but there has been an explosion in microRNA analysis in the cell-free DNA fraction or RNA fraction, and that is very stable, and we think that that can provide some valuable information. Yeah.
A couple of questions. Have you looked at any oropharyngeal or airway malignancy cases?
If you notice, lung cancer wasn't on there.
I did.
And one of the challenges with lung cancer is getting tissue. Most lung cancer is diagnosed with an FNA. Early stage lung cancers you can get from, which are rare, resection. So we wanted tumors where we had the tissue and we had the plasma. And that's one of the main reasons that we didn't go there. There have been recent studies from other groups looking at circulating tumor DNA in lung cancer, and it's there. We just couldn't do these type of studies for them.
Second question. I know it wasn't the bulk of your talk, but you mentioned fetal DNA.
Yeah.
Whatever. Do you know how long, or does someone know how long that fetal DNA persists in the mom after?
It's 2 hours.
delivery?
Half-life is 2 hours.
Is that all?
So you know the studies.
Is there no chimerism of the kid's cells in the mom?
No, there's some suggestion that there may be cellular chimerism,
Yes.
that fetal DNA persists in the circulation. But there's very elegant work by Dennis Lowe where and these mothers must have been saints, they allowed them to draw blood every 15 minutes after delivering the baby. And they precisely saw the half-life about 2.5 hours in these patients.
Cool. Thanks.
Yeah.
So do you have an estimate of what's the minimum number of tumor cells you require to detect, tumor mutations either in plasma or tissue?
So we've done this in colon cancer, and we've done it by imaging, so it's imperfect. But we think it's about 1 billion cells. It's about 0.5 centimeter to 1 centimeter.
So how much plasma do you actually use to extract the
About 2 cc's. And this is where these are some arguments we have with some collaborators sometimes. Everyone's driving at developing a more sensitive technology. But in 1 mil of plasma, you have about 2,000 genome equivalents. 1 in 10,000 you don't gain anything there. Even with 2 cc's, you have 4,000 genome equivalents. The best sensitivity you can get there is maybe 1 fragment in 4,000. But it's typically 2 fragments in 4,000 if you want to be statistically robust. So you have to collect 5 to 10 cc's to get between 10 and 20,000 fragments for a technology that can detect something with sensitivity of 1 to 10,000, 1 to 20,000 to even be useful. So we've seen a drive to develop a more sensitive technology, but we think we've hit some biological limits at this point. Yeah.
And the circulating DNA, does it travel in a microsome or something that you could actually plasma freeze off or something?
So there are a lot of people who think they're within exosomes, or small vesicles that butt off the cells. But we haven't found that to be true, and our extraction method takes everything into account. We've done some studies where we've done ultracentrifugation, collected these exosomes, and there's a really small amount of DNA there, much less than in the total circulating DNA fraction. So we don't think there's an enrichment there. There might be in certain tumor types that release more exosomes. I'm not sure. Yeah.
So, comment and a question.
Mm-hmm.
The one comment is that these nucleic acids, certainly microRNAs, have been shown to associate with albumin, for
Right.
example, and albumin is a sink. I'm wondering if you considered looking at that for earlier detection. That would increase your sensitivity. If what you're looking at is an overload, and really what's the early stuff is being sucked up, associated with circulating proteins. Have you thought about that?
Yeah, so we've done immunoprecipitation studies where we've actually immunoprecipitated the nucleosome, to see if we could enrich or capture cell DNA that we're not capturing by our extraction method. But the kit we use that we apply to the plasma or serum, it's a QIAGEN kit, so I don't know the detergent they use there. I suspect it's like an NP40 detergent and proteinase K, so you do get degradation of all that. But you're right, we may be able to enrich by looking at other larger proteins that these associate with.
So we have a new faculty member, actually lives in Duluth, Lynn Venus, who does a lot of this. I don't know if you've heard of her, but she's worth talking to.
Yeah, no, that it's definitely interesting, because getting from that threshold to 50 to 40% for early stage disease higher, we've got to think of ways to do that. And
just a second question, at the other end of the body from what Leo was asking, in terms of prostate cancer, urologic cancers, are these tests available for urine? And is there any utility potential?
so about a year ago,
Well, it's more important for some of us. It's more important.
so a year ago we looked at, we found a bunch of TERT mutations in prostate and bladder cancer in the promoter region. And then recently we've been looking in the urine for bladder cancer and prostate cancer and kidney cancer. In bladder cancer, it's abundant. In prostate cancer, we don't know yet. And in kidney cancer, we'll know soon.
Okay. Focus on the prostate.
Any other questions?
All right, thank you.